Thank you, Martin, for that very kind introduction. It's a great honor and a great privilege for me to have an opportunity this morning to tell you a little bit about what my students and I do here at the Indian Institute of Science in Bangalore. I have titled my talk as "Can we understand an insect society and why should we care?" Notice that there are two questions in the title, and I will try and answer both of these questions. But let me begin by introducing you to the absolutely fascinating world of insect societies. Many of us know, but have not reflected, I think, in sufficient detail, that many species of insects organize themselves into societies, much like our own society. And these insect societies are very sophisticated in their social organization. They have excellent social coordination, communication, division of labor, leadership, all of these qualities. And in some ways, I would venture to say that they surpass human societies. The example that almost everybody is familiar with, of course, is the honeybee society. As all of us know, honeybees live in large populous colonies consisting of tens of thousands of bees. But in each colony, there is invariably one single large fertile female bee, which we refer to as the queen bee. She's the only one who reproduces. And the rest of the colony really work for her welfare and the welfare of her offspring. I might mention in passing that honeybees colonies also have a few males. They only deserve to be mentioned in passing because the males are incurably lazy and do nothing for the society. But that's another story. My students and I study another insect society, which is actually a social wasp. And we study this because it has many attractive features as a model system. Its colonies are small, unlike the honeybees. A colony may consist of just 20 or 30 individuals. They build small colonies, and they're much easier to study, but most important of all, they're abundant all around here. There are some in this hotel, the buildings, they build their nests in gardens, in trees, in buildings, they're very, very common and very convenient to study. But an important distinguishing feature of this society compared to the honeybee society is that unlike the honeybee society, this society does not have a single large fertile queen. It has a single fertile queen, but not large. Indeed, the queen of this colony, and there's only one per colony, is indistinguishable from the rest of the workers. And she is a queen only for a while. She can be replaced at any time by one of her workers. These workers in the past actually used to be called slaves. So she can be replaced by her slave, who will then become the next queen. I take the questions in my title seriously. Can we really understand an insect society? And I think we should not be too cocksure about this. I will begin with a statement made by a Greek philosopher, Heraclitus, who said, "Nature loves to hide." And I think we should all pay attention to this, because this will then give us the much needed caution when we do research and when we think, as soon as we find something, that we've really understood nature. So this quotation guides our research and gives us the required caution. However, we are also very fond of another statement made by another philosopher, an English philosopher, Sir Francis Bacon, who said nature does not unveil its secrets except under the torture of experiments. This gives us hope, because we love to do experiments. So armed with caution and hope, we study this insect society. Its technical name is Rhopalidia marginata, and as I said, it's a social wasp. Another special thing about social wasps is that social wasps are called paper wasps. They are called paper wasps because they build their nest from paper, not from wax as honeybees do, or leaves that ants do, or or soil as termites do, but indeed from paper. You might ask, where do they get their paper from? They manufacture paper. How do they manufacture paper? In precisely the same way that we manufacture paper. They scrape cellulose fibers from plants, chew it up, add some chemicals, make it into a fine pulp, spread it into a thin layer and dry it. And it is indeed paper. You can write it. The whole nest is built from paper. The second question in my title is, why should we care? Indeed, why should we study them? Now, those of us who study insect societies, and there are only a few thousand of us in the world, we have several different kinds of motivations. Some of us are motivated by simply using knowledge from the insect to make money. Many people have used how these insects work. Social insects are networks of individuals, decentralized, self-organized, bottom-up creatures. And that's why they can achieve all those fantastic things. And people have used the methods that insects use to organize themselves to organize human societies, to organize various things in human societies. There is, in fact, a whole branch of computer science which is inspired by social insects. It is called ant-inspired algorithms. It is even true that the Southwest Airlines, for example, uses ant-inspired algorithms for cargo transport. Telecommunications industry uses ant-inspired algorithms. The internet traffic is now being inspired by ant algorithms. There are others who make money by using the fact that honeybees pollinate our crops, our orchards, and honeybees secrete a suite of chemicals through which she attracts her workers and makes sure they're close to her. People have identified these chemicals, synthesized these chemicals, and they spray it on the orchard, and a passing worker thinks, why is my queen here? And she goes to the orchard and she inadvertently pollinates these crops. So people have made a lot of money by doing this. Honeybees, insect societies in general, are an extremely interesting model system for evolutionary biologists. And this is because they at least seem to defy natural selection. They seem to defy Darwin, because most of the individuals in these societies are sterile. They do not reproduce, and yet they are perfectly adapted to the task they perform. And Darwin asked, how is it possible? And this is a question that's still being discussed and debated. At least for the portion of our work that I have chosen to discuss today, the motivation is very, very different. And let me explain what this motivation is. Why do you study social wasps? People often ask me. For the same reason that an anthropologist studies humans is my answer. Anthropologists can offer us a glimpse into the lives and mores of primitive and exotic human societies. Biologists can do much more. They can offer us insights from a whole range of animal societies with millions of years of evolutionary history. And those of us who study insect societies can hope to harness wisdom from an altogether different sub-kingdom of life. Wisdom is the operative word here, not dollars or pounds. Many people ask me, how do you actually study them? These wasps, as I said, build their nests everywhere. We can study them where they occur. We also discovered that we can transplant them, we can literally fool them by moving them at night to our laboratory, and then they get used to our laboratory, they live in our laboratory, and we study them there. And when we transplant them to our laboratory, we do not have doors, walls, or windows in the laboratory. We just have a wire mesh screen, so they actually go out and come back. They go bring their own food, they bring their friends, they leave, they join. It's almost a natural situation in which they work. In addition, we are able to put little spots of color paint on the different parts of the body and therefore uniquely identify every individual. Thousands of individuals are identified in this fashion. They all have a little name which is in our computer file, and we can build up dossiers on each wasp because it's individually identified. In the laboratory, we can put them in little plastic boxes or in cages of this kind, and we can stack lots of cages in this laboratory, which, as I said, does not have walls but only a wire mesh. Now, the aspect of the work that I'm going to try and discuss today has to do with the two aspects of their behavior, conflict and cooperation. It is remarkable that these insects show both conflict and cooperation. In fact, cooperation and conflict are finely balanced, and that's what I want to talk about. But I also have some collateral messages that I want to give through this talk. And one of the important messages that I want to give is my belief that the process of science is as important as the product. We often discuss only the product of our research. We only convey when we give lectures, when we write papers, we neglect the process by which it is done. And I think this is a great shame, because not only is the process equally, perhaps more interesting, but it's important to convey the process. The product of my research may be of interest only to a small section of people, but the process may be of interest to a much wider section of people who may be able to use this process to do other things. And I think it's very important to speak and talk and convey the process of science. A word about how our research progresses. I and my students, we love these WASPs. They're great fun to watch. So we can watch them just for pleasure. And we often watch them for pleasure. And when we watch them for pleasure, some question occurs in our mind. And then we are reminded of what Sir Francis Bacon said, and we design an experiment to try and answer this question. And if we succeed in answering this question, the joy of answering this question is usually very short-lived, because another question appears in our mind saying, how can this be true? And then we go back to Francis Bacon and we design another experiment, and we continue in this process. So it's a series of questions and answers. And since I want to talk to you about the process, this talk is going to be a series of questions and answers. So I'm going to take a few examples of the questions we asked, the experiments we designed, and the answers we produced, and why our joy at that answer was short-lived. What was the next question? When I first started studying these wasps, I was struck by the fact that the queen doesn't look different from anybody else. So what I did was that I wrote down in plain English all the different things that these wasps do, all their behaviors in ordinary plain English. And I came up with a list of about 100 different things they do. And I asked, does the queen do anything different from the others? And the answer was no, except that she's the only one who lays eggs. Most wasps seem to do most of these things. So I realized that the difference between the queens and workers must be in the quantitative nature, in the actual amounts of time they spend in doing different things. So I developed methods by which I could calculate how each wasp spent its time or allocated its time to each different behavior. So I had a large numerical data matrix, rows of wasps, columns of behaviors, and each cell was the proportion of time each wasp spent in each behavior. This data matrix was ideally suited for multivariate statistical analysis. So we did some principal components analysis and some cluster analysis, and we came to the rather surprising result that every colony consists of three kinds of individuals, which we call sitters, fighters, and foragers. I just forgot to say that I will talk both about conflict and cooperation. My title was Conflict and Cooperation, but I'd like to begin with stories of cooperation and come to conflict later. So when we had this classification of sitters, fighters, and foragers, then we post facto asked, where is the queen in this scheme of things? Is she a sitter? Is she a fighter? Is she a forager? Common sense told us, actually convinced us, that she must be a fighter. Because in other such species which had been studied, where the queen is not a morphologically differentiated individual, queens are highly aggressive, highly interactive individuals, and they literally rule by physical force. They bite the workers, they chase the workers, they harass the workers, and through such physical aggression, they make sure that they are not overthrown by the workers. They also make sure that the workers actually work. So our queen must be a fighter. But of course, she is not a fighter. In colony after colony, we find that our queen is a lazy sitter. This is very unsettling, because if indeed our queen is such a meek, docile sitter, how does she become a queen in the first place? Why do the workers accept her as a queen? Because in species where the queen is very aggressive, they have no choice. So we said, we must design an experiment. So we designed an experiment where we said, we want to witness the process of the making of a new queen. And how do you make a new queen? Simple, remove the old queen. So we decided to do the experiment where we study all the wasps in a colony, queen and workers, for several hours, determine the proportion of time each of them spends in each behavior, and then experimentally remove the queen and study the rest of the colony, the rest of the workers, again calculate how now do they spend their time in different behaviors. When we first did this experiment, at the end of the second day, we had the queen still sitting in a vial. We had given her a drop of honey. She was quite happy, and we wondered what to do with this queen. And then we thought, why not put her back into the colony? Because we didn't know what would happen. We put her back in the colony. She just went back to work to be a queen as if nothing had happened. And therefore, we now studied the wasp on the third day with the queen reintroduced. So you have day one with queen, without queen, and with the queen returned. And when we removed the queen, something spectacular happened. This species is, even though the queen is very meek and docile, has some amount of aggressive behavior. These are called dominance-subordinate interactions, biting, chasing. Now, there is a certain amount of this, but this is a reasonably peaceful society. The moment we remove the queen, this reasonably peaceful society becomes an extremely aggressive society. We have measured the change in aggression. The levels of aggression in the colony increase sometimes by 2-fold, sometimes 10-fold, 30-fold. We have even recorded a 40-fold increase in the aggression as soon as you remove the queen. What is even more remarkable is that all of this new aggression is shown by one individual. One worker becomes hyper-aggressive. This is what data would look like. Day one, there is almost no aggression. Day two, one individual becomes hyper-aggressive. Interestingly, when you return the queen, she drops her aggression and goes back to work, respecting the rights of the queen. So the answer to our question is, in Rogladia marginata, queens begin their careers as very aggressive individuals and only later become meek and docile. So that explains why they are accepted in the first place. But this is not satisfactory, because if that is true, how does she inhibit worker reproduction? Because she may be a queen for the next 8 months, and she has been aggressive only for 1 week. How does she make sure that 1 month later, somebody does not challenge her and overthrow her? So we decided to do another experiment to answer these questions. We hypothesized that, like the honeybee queen, this queen also may have a certain chemicals, these are called pheromones, which she may produce, and through this, she may actually physiologically suppress reproduction by the workers. She doesn't have to go and chase them, and she doesn't have to keep biting them. So in order to test this possibility, we decided a different experiment. Here what we did is we brought a whole colony to the laboratory, put it in a cage, studied all the wasps on day one, and on day two, instead of removing the queen, we removed all the wasps from the nest. We took a blade and cut the nest in half with roughly equal brood and cells on either side. And then we took each wasp, tossed a coin, and decided whether she goes to the left or to the right. Next wasp, toss a coin, heads you go left, tails you go right. Finally, queen, heads you go left, tails you go right. So we have one fraction, half the workers and the queen, the other fraction, other fragment, separated by wire mesh with half the workers. And on day 3, we simply moved the queen from where she was to the opposite side. Now, why did we do this? Our predictions were as follows. If the queen pheromone is volatile, both nest fragments should behave like queen right colonies. That is, the wasps on this side should have no difficulty in realizing that there is a queen across the mesh, because they should be able to smell her. And they should behave as if nothing happened. And same here. But if the queen pheromone is non-volatile, the queen-less fragment should behave like a queen-less colony, because these wasps now think they don't have a queen. And what does behave like a queen-less colony mean? A hyper-aggressive individual. Now, I forgot to tell you that if we do not return the queen, the hyper-aggressive queen will actually go on to become the next queen. This is, in fact, the process of queen formation. In a week's time, she will lose all her aggression and start laying eggs. So we expected this to happen. And in all the experiments we have done, when this slide was prepared, we had done this 24 times. Now we must have done it 100 times. And in all experiments, prediction 2 is upheld. The data would look something like this. Day 1, nobody is aggressive. Day 2, one individual, we call her potential queen 1, on the queen-less side becomes aggressive. But when the queen is moved to her side, she loses her aggression. A different individual who showed no aggression when she was with the queen now suddenly becomes aggressive, because the queen is no longer on her side. And therefore, we believe that in Rogladia marginata, queens appear to use non-volatile pheromones to inhibit worker reproduction. How is this possible? Because if this is true, how does she regulate the non-reproductive activities of workers? Aggressive queens in other species use aggression not only to prevent others from becoming queens, but to make sure they're working. If they stop working, they will go and bite them. Now, you can perhaps produce a chemical which will physiologically suppress the ovaries of your workers. But you can't produce a chemical and say, go and work. Why would anybody obey you? So there must be something else. So we said we must design another experiment. Here we use the same experiment where we remove the queen, but we focus on work. And we took two examples of work, two perhaps most important work that the workers do. One is to bring food, and the other is to feed the babies, feed the larvae. We measured this carefully in the presence of the queen and in the absence of the queen to understand how the queen might regulate these activities. And to our great surprise, we found that the queen does not do any such thing. The workers don't need to be told. They bring food day one with queen, without queen, queen return at exactly the same rates. They feed larvae at statistically indistinguishable rates. They don't need a queen. They know what to do, and they keep doing that. And the queen does not regulate foraging by the workers. We have since actually shown that workers themselves regulate their own foraging in a decentralized, self-organizing manner. And I'll show you a little bit of data. In the interest of time, I won't go to the details, but I'll show you a little bit of data that this might be true. And this will come in the form of an answer to another question. Now, our next question is, what is the function of this low level of aggression that happens in colonies? There's a low level of dominance, subordinate interaction that happens. What is the function of this? In other species, the function postulated is that amongst the workers, the one who is most dominant is going to be the next queen. That doesn't seem to work here. So why do they do this in the first place? Now, we try to understand what this is, and by what we generally call a fishing expedition. We just looked at all possibilities, and we found one interesting correlation. There is a weak but statistically significant positive correlation between the fraction of dominance a worker receives and how hard she works. So the more you beat me up, or somebody beats me up, the harder I work. So we had a hypothesis that this low level of aggression in normal colonies, in queen right colonies, is actually used by the workers to self-organize regulation of each other's work. But this is just a correlation, and one should not infer cause-effect relations or correlations, as all of you know. So we designed experiments where we are more confident. We decided if this is true, if this is indeed true, then of course, excess feeding should reduce dominance behavior. If nobody is hungry, then they should not show aggression. Now, we actually designed experiments where we made sure nobody is hungry. This was done by one of my students who said, I will hand feed the wasp. She would take a piece of caterpillar at the tip of a forceps, and she would sit in front of the colony. The wasp would initially be frightened, but after a while they would come, grab that piece of caterpillar, run away and eat it, and she would provide one more. Then they would take that and she would give one more. They would take that and she would give one more. And then they would start feeding their larvae, and finally they didn't want any more food. They were completely satiated. Then you make observations. And she was able to show that bringing food came down if you fed them, feeding larvae came down, their dominance behavior came down. Indeed, the dominance specifically that they showed towards foragers came down significantly when they were all fed. Another student did the converse experiment. She showed that starvation increases dominance behavior. This was somewhat easier to do because you put the wasp in a cage, close the door and say no food today. And when you do that, you can actually show that the dominance behavior actually goes up in the absence of food, and it is not random aggression. It is not just stress and frustration, but the very wasps that brought food on the previous day were targeted. That is, those who were known to bring food yesterday were targeted today when everybody was hungry. And when the door opened, they of course started bringing food. So we think that worker dominance behavior, the low level of aggression that you see, is in fact used for the decentralized self-regulation of foraging by the workers. There is, of course, another question that needs to be answered, and everybody asks this question. You remove the queen and you say one individual becomes hyper-aggressive, and if you don't return the queen, she will become the next queen. Who is this individual? Who is the successor? And as humans, we would be terribly interested in knowing who is the next one in charge, and how is and when is the queen here decided. Now here I will tell you a somewhat different story. People, of course, always ask this question, and our answer was, we don't seem to know. It's not easy to tell. We can't tell. It seems to be confusing. It seems to be random. It seems to be complicated. Now, after a while, when we were asked this type of question many times, this was not satisfactory. We said we must do something about it. We must design an experiment to try and predict the potential queen even before the old queen is removed. Now, the best way to solve a hard problem is to tell your next PhD student that if you solve this problem, you'll get a PhD. So my next student was told, you have to predict who the successor to the queen is before the queen is removed, and you will get a PhD. Now, this poor girl spent 5 years working very hard. She did a lot of experiments, produced a lot of data, but she threw up her hands and said, I'm sorry, I cannot predict the potential queen. She got her PhD all right, but she could not predict it. Just to summarize with one slide, what she showed is that the potential queen and the workers are completely mixed up. The potential queen is not unique by any criterion, dominance rank, or any other behavior. Indeed, not even in the small amount of ovarian development that she has related to other ones. None of these things seem to allow us to predict who is successor. There was a time when I used to be embarrassed to say, no, I don't know. But believe me, now I sort of feel that these wasps hold some secret that I don't know. It's very charming. I really don't know what it is, but I love my wasp so much that I'm really quite happy to say that I really don't know what it is. But the next student who came to my lab said, we cannot predict, but I want to ask a more difficult question. We don't know who the successor is. Do the wasps know who the successor is? This does not seem like a scientific question. It does not seem like a question that can be answered by science. How would you know the mind of a wasp? But she was persistent, and I might identify her. She's in fact in the audience. Dr. Anindita Bhadra, who is now a distinguished scientist in her own right, did these experiments. So we tried to design an experiment by which you can actually answer this question. We spent a lot of time discussing, doing experiments, discarding these experiments because we ourselves found a flaw in the logic, and going back to the drawing board. Finally, I think Anindita hit upon a design by which we can actually answer this question. So the question is it possible that there is a hair designate, even though we cannot identify her? Do the other wasps know who this hair designate is? And I will describe the design of this experiment using a little animation. The experiment is tedious to do, but not difficult to understand. We use the same design where we put a nest in a cage and put a wire mesh in between. The queen and half the workers on one side and the remaining workers on the other side. Now, just by chance alone, so let us assume that there is a hair designate. And let us assume that all the wasps know who their hair designate is. We are now examining the consequences of this assumption. If this is true, what would we find? We would find that the hair designate, by chance alone, might have ended up on the opposite side of the queen. And if that happens, then of course she will immediately become a potential queen. No problem. But if she's indeed the hair designate for the whole colony and not just for her side, so we move her to the opposite side and she should be acceptable to the wasps here, we move her back here, she should have no problem. However, in about half, this can only happen in about half the experiment, because in the other half of the experiment, just by chance alone, the unfortunate hair designate would end up along with the queen and she can't do anything about it. Now, the best individual on that side will become the potential queen, but then we will exchange her position. We will bring her to this side, face to face with the true hair designate. And now all hell should break loose. And this individual should not be the potential queen anymore. This individual should become the potential queen because she's a true hair designate. But that's not the end of the story. The acid test for this individual is we move her to the other side and she should be acceptable to the opposite side. So our summary predictions are the first potential queen should be unacceptable to the opposite side in about half the experiments, and a second one should emerge. But the second one should be acceptable to both sides. There should be never be a third contender to the throne, as it were, if you cut the nest in two parts. Anindita did 8 of these very difficult experiments. They were very difficult on her, but I can assure you they were much more difficult on the wasps. And she managed to do 8 experiments. In 3 experiments, she found that the first individual was aggressive on both sides. And in 5 experiments, the first individual lost her aggression. The second individual was aggressive on both sides. So both predictions were upheld. A potential queen to emerge in about 50% of the cases. No PQ3 ever emerged. Both. So we think there is a hair designate, but she's a cryptic hair designate because we don't know who she is. Now, the conclusions of this paper is interesting. People always asked us, do you know who the successor is? And we always used to say no. But now we got a great kick in saying, we don't know, but we know that the wasps know. And I'm proud to say that we convinced the editorial journal to keep this title, We Know That the Wasps Know. They're a cryptic successor to the... But the real message of this experiment is not conveyed in the numbers, is not conveyed in the data. It turned out, it's remarkable, that the second potential queen was never challenged. She did not receive a single act of aggression by the potential queen one or by any of the other workers, either when she emerged on this side or she went to the other side. And that is why we think that the hair designate was obviously known and acceptable to all the wasps, including the potential queen one. And that's why we think that they know who this is. So there is a hair designate, even though we cannot identify her in the presence of the queen, the wasps seem to know who she is. The next one who came and said, is there just one hair designate or is there a line of them? So I do the experiment. So he removes the queen, finds a potential queen. He can identify her in half an hour to 1 hour. Remove her. There's a next hyper-aggressive individual. You remove her, there is another one. And he actually showed that in the normal colony, nobody shows aggression. Remove the queen, one and only one individual. Remove her, one and only one individual. Remove her, one and only one. And you can go on like this. And in fact, there is a line of these, and they all show aggression, but none of them are challenged. They are unchallenged, but only one individual stands up. And therefore, we think that there is, in fact, a queue of potential queens, successors, pre-designated in the colony. Now, the question is, if there is so much cooperation, there's so much peace in the colony, where is the conflict? We were hoping to see conflict when we brought the potential queen one with the true hair designate. But no, they are so civilized, the potential queen one just went back to work as if she knew that it's not her turn yet. But you can see conflict. If you want to see conflict, you just have to bring two wasps from two different colonies face to face, and you will see that there is conflict. So the question we ask is, can the wasp discriminate nest mates from non-nest mates? Who belongs to my nest and who does not belong to my nest? We designed experiments where we put 3 wasps in the cage, 2 of them are nest mates, 1 is a non-nest mate. We studied their behaviors, we created an index of tolerance, and we were able to show that nest mate discrimination, I won't go into the statistical details, nest mate discrimination is well developed. Conflict with outsiders is indeed possible. Rhoplidia marginata wasps have a well-developed mechanism of nestmate discrimination. They are very tolerant of nestmates, but highly intolerant of non-nestmates, even in isolation, even under laboratory conditions. Next question is, how do the wasps treat intruders from other colonies? Occasionally, intruders come in. So we did the following experiment. We took all the wasps from one colony and introduced them to the cage containing another colony. So there were resident wasps and there were alien wasps. And believe me, the response of the residents to the aliens was very nuanced, was very measured. It was not a simple response. Indeed, what happened was that when the members of one colony were introduced into the cage of another colony, the young alien workers were freely admitted into the colony. Come and join, no problem. Older alien workers were allowed to live, but in some corner of the cage. As long as you are far away from my nest, no problem. The alien queen was located, attacked, and dismembered wherever she was. So the wasps are able to recognize whether you belong to my nest or not, and even if you don't belong to my nest, what kind of an individual are you? Are you young? Are you old? Are you a worker? Are you a queen? They're able to make this recognition. Now, if young wasps can be accepted into the colony, can the wasps keep track of these young intruders who sneak into the colony? So we did another experiment, and we found that no. We found that intra-colony discrimination is undeveloped. So once I admit you to my colony, then you become a card-carrying member of my colony. Then I can no longer tell that you don't belong to us. You can no longer tell that you don't belong to us. So in fact, there is no evidence of intra-colony discrimination. Young alien workers who are accepted into foreign colonies become completely integrated into their foster colonies and may even go on to become future queens. We have determined that the probability that an alien will become the next queen is proportional to the number of aliens in the colony. That's all that matters. As long as you are in abundance, you can take on the next. So you can see there is indeed conflict and there is cooperation. The wasps have a great propensity to have conflict with outsiders and an equally great propensity to cooperate with insiders. I've shown you that they bend over backwards to be nice to each other and cooperate with each other and work even when they are not told to work, when they're in the colony, but outsiders they are not. From an evolutionary point of view, conflict with outsiders, of course, is easier to explain than cooperation. The extreme cooperation inside is actually harder to explain from an evolutionary point of view. But if you think about it, conflict with outsiders is really not of much use unless one can combine it with conflict with insiders. There's no point having conflict with outsiders if you also have a conflict with insiders. Then it doesn't do you any good. It is this dual strategy, I believe, this ability to tread a fine balance between cooperation and conflict that I think accounts for the huge ecological success of insect societies on this globe. Insect societies are some of the most ecologically successful. In many forests, in terms of biomass, insect societies account for more than even vertebrates. And that's why we call them superorganisms. But let me come back to my initial statement that I study these wasps just like an anthropologist studies human societies. And I cannot refrain from reflecting on human societies. And I cannot help saying, we humans are the same, aren't we? And this is my final philosopher's quotation. This is Voltaire, who said, "It is lamentable that to be a good patriot, one must become the enemy of the rest of mankind." So what is my overall take on studying these wasps? I certainly do not think that we should imitate insect or any animal societies blindly. But I certainly think that they can hold a mirror to us and offer us a means to reflect on our own society and learn more about ourselves. It's only when you look at someone else that you understand yourself better. You go to your neighbor's house, you look at how they have organized furniture in their drawing room, and you realize that you have done a much better job than they have. You might go to your neighbor's house, see how they treat their children, and say to yourself, I hope I don't treat my children that way. You go to a foreign country and you realize how you behave. You always need a mirror to understand yourself, to reflect on what we do, because we do things automatically. And I think animal societies, insect societies, give this mirror to us. And they're all over the place. They have lived here for millions of years before us. And I think we should have the modesty to look at them, to study them, to learn, and to get wisdom from them. I want to end by making another different kind of message. Now, I'm sure you would have noticed that everything that I've said required no equipment, no sophisticated laboratory, hardly any chemicals, very little money. And this is deliberate. I really enjoy working in an environment where the only limiting factor is my brain. Nothing else should be the rate limiting step. Rate limiting step is my ability to think. But people often ask me, where are the sophisticated instruments in your laboratory? I have lots of them. They're here. They're my stick. So most of the time we spend talking, sparring matches, thinking, discussing, doing an experiment, coming back, throwing that experiment away, doing another experiment. And this kind of science also gives hope that even those of us who are not so well endowed in terms of a laboratory, in terms of money, in terms of grants, we can actually do very interesting science, which gives us great satisfaction. And that's the message I want to leave you with. Thank you.
Thank you, Professor Gadagkar, for a beautiful exposition of the social wasp and the way you developed your entire story. We can have a couple of questions. Yeah, please, Arun. Yeah.
Thank you very much for an interesting talk. My name is Raghavendra from Jamaica. Is there anything special about these wasps to identify and remove waste from their colonies?
Yes, they do. Of course, this is much more developed in societies which have very large colonies and which live in closed nests. The advantage these wasps have is there are a few of them, so there's relatively less waste, and they're on a small open colony on a vertical surface. Most waste falls off with the help of gravity, but yes, they do. If there are dead wasps, dead larvae, parasites, they're able to clean. And they're constantly cleaning and nibbling the paper with which the nest is made to remove parasites. And if they don't do that, they will get infected. There are many parasites, including fungi, which will infect the nest, but for the constant cleaning process that they do.
Yeah, please go ahead.
Thank you. My name is Muregi from Kenya. Thank you for the exciting talk.
Thank you.
I've noted your work is behavioral observation or just simple observations. I'm wondering whether you have explored some chemicals maybe we don't know about, physiological level, what is happening. Maybe other than aggression and whatever, there is that chemical probably they can detect to know this could be the potential queen and whatever, and maybe we don't know about it yet.
Absolutely, absolutely. Let me tell you as briefly as I can. So when we found that this chemical is non-volatile, the question we asked is, how do the workers receive it? How do they perceive it? The obvious answer was they must be going and interacting with the queen, they must be touching the queen, and they must be therefore acquiring this chemical. We did a number of experiments and we showed that the rate at which they interact with the queen, because the queen is a very non-interactive, shy individual, that rate does not allow us to explain the rapidity with which the potential queen realizes that there is no queen and therefore it's my turn. So it doesn't work that way. So how does she do it? Then we found, in fact one of my students found, that the queen rubs her abdomen on the nest as she walks around. And then we postulated that maybe she rubs the chemical on the nest, and that chemical the workers get from the nest. All you have to do is to turn the wasp upside down and see what's there. And indeed there's a hole there. And there's a duct there which leads to a gland. So we took out that gland, this is called the diffuse gland. We made an extract of that gland. And we said, if our hypothesis is correct, then I remove the queen, I have a hyperaggressive individual. Now instead of returning the queen, I will apply this chemical on the nest, this crude extract on the diffuse gland, and the worker should drop her aggression. And indeed she does. So the diffuse gland contains the chemicals. Then of course we put the extract into a GCMS machine. We looked at it. There are 24 to 25 different compounds. No compound is uniquely present in the queen compared to the workers. No compound is uniquely absent in the queen compared to the workers. It's the cocktail, it's the ratios that matter. We've even shown that on day 0, when the queen is still present, the cocktail of the potential queen is identical to the cocktail of a typical worker. In 1 week's time, if you remove the queen, the cocktail of the worker changes in multidimensional space and becomes indistinguishable from the cocktail of a typical queen 5 days later. There's actually a change in the composition of this. So we are studying the chemistry.
Please, go ahead.
Excellent presentation. My name is Jaya from University of West Indies. Even though I'm not a social biologist, I got excited after hearing your lecture. The question is, every individual, every member of the colony has has an opportunity to become a queen, or is there any kind of prereq? Number 2 is, you cut the colony into half,
yes,
just by longitudinally. Did you try it vertically? I mean transfers? Because in the case of honeybees, I read some time ago that the level of pheromone is always less in the bottom half. And that's how they can just flee away and then make a colony. I don't know how far it's true.
Yeah, in the case of queens, you're right. And that's because the queen spends most of her time only in one part of the colony. And there, the queen actually produces volatiles. So you can actually show there is a gradient of these chemicals. Here, the colonies are very small, and the queen is all over the place. And remember, it's non-volatile. In fact, we have shown statistically that the queen rubs her abdomen all over the nest. We actually divided the nest into different quadrants, and we measured quantitatively how many times she rubs in this quadrant, how many in this, how many in this. So we have statistical data to show that she does this uniformly throughout the nest. And also in these kinds of wasps, vertical and horizontal doesn't make any difference, because sometimes the wasp nests are built like this, sometimes like this, sometimes like this. So they're built in all kinds of configurations. So we don't think that vertical or horizontal would really make any difference.
And any member can become a queen?
Well, that's complicated to answer. Now, I showed you this experiment, you go up to 5. Now, first of all, even if any member can become, you have to wait your turn. And the wasps, for some inexplicable reason, wait their turn in a very civilized way and do not jump the queue. That is really a paradox of your answer. Now, whether this queue goes on to the last wasp is very hard to test, because the wasps get tired of you if you do this experiment too often. So my students said, say, I've shown it for 5 successors, and that proves that the proof of principle, I don't have to go further. So there are technical reasons showing even the very last one can do that.
Thank
but
you.
It's very likely.
Please go ahead. Give the mic.
Thank you very much for the interesting presentation, Prof. My name is Mgiti Dlamini. I'm from the University of Swaziland. I'm very much encouraged by the conclusion you made, Professor, to say that really equipment or facilities should not actually hold us back. I know that quite a number of us here who have studied abroad, for example, in Europe, USA, elsewhere, have actually used very complicated equipment where they were. But when they returned home, they actually found that there is nothing to use. But like you say, what is important is the human resource. What is important is actually using the knowledge we gained during our studies and converting it into something useful. In other words, we don't have to be held back by the complicated equipment. We should find something else to do which is related to what we're doing elsewhere. I'm also encouraged by the comment made earlier by Professor Rao, actually, who took us through his history. Thank you very much.
Thank you for your comment. Can I have a last question?
Yeah, I just want to know the time scales you're operating, like you have done an experiment.
Get the mic.
Sorry. I was just curious, excuse my ignorance. The time scale, you say day 1, day 2, day 3 and things like that. What happens if the temporal scale is changed? How long do they remember? I mean, I'm just trying to find out if humans are anyway superior to these insects.
Well, let me tell you also a little bit about how we do this. People often ask me, you return the queen 24 hours later, she's accepted, the potential queen goes back to work. What if you return her a little bit later? This question. And every time people ask me this question, I said, this experiment can be done, but I'm not motivated to do it. Because what will it tell me? It will tell me that after X days, the old queen will no longer be accepted. That X may be some number, 7 days, 6 days, 5 days, 20 days. What will it tell me? So I actually refused to do that experiment. Even some of my students wanted to do that. I said, but give me a reason to do that experiment. Now we have done that experiment because we found a good reason to do the experiment. Once we found that there are these pheromones and that there is cocktail and this cocktail changes, we said we can be the first people in the world to behaviorally titrate a chemical. Because on the day the old queen is rejected, the potential queen says, who are you? On that day, the pheromone composition of the old queen and new queen should be identical. So behaviorally, we can actually titrate not just one chemical, but a cocktail. So then when we got that motivation, we have done that experiment. And that seems to be approximately around 48 days. So now we are doing 12-hour intervals. So we're actually measuring how long does it take for the old queen to become new. I told you at the end of 5 days, the potential queen becomes indistinguishable from a typical queen. But she doesn't have to wait that long to be rejected. She just has to be as good as the potential queen, who is not yet like a typical queen. She is coming up and she is going down. So we are trying to find this end point in our titration, and we are now doing these experiments.
Thank you, Professor Gadagkar. Let me present a memento.
Thank you. Thank you.